Hi, this is the part of the program www.resetonline.org, hands-on elementary school science experiments. This particular experiment involves a fish, and I received a note from one of my students after the completion of the experiment. Thank you for bringing the fish. It was my favorite experiment, getting to feed them every week. It's sad that our fish died, but we had a good time anyway. Just make that point that working with these young students is a lot of fun. We have to introduce vocabulary at the start of each session, and this session has to do with growing things. The vocabulary is energy, nutrients, photosynthesis, scales, and calibrate. I do this experiment in 2 separate sessions, 6 to 8 weeks apart, so that we can measure the difference in the biomass of the plants at the beginning and at the end. We start by talking about the requirements of living things, and it's good to ask the students what do living things need, plants and animals, because it'll give you a gauge for how much they already know. So we get into they need energy, talk about the fact that plants get their energy from the sun or from light, and that animals get their energy from the food. Talk about nutrients, that plants get theirs through their roots from nutrients in the soil, or animals get their nutrients also through their food, the same as their energy. And then, of course, water and oxygen often comes up in that discussion as well. We have the students weigh the plants at the beginning of the experiment and at the end in order to measure the change in the biomass weight. I teach the students how to read these scales, diet scales that I get from Bed Bath & Beyond. And I start by giving them a handout, which has sketches of the dial on paper with the needles pointing to different points, and teach them how to read the face of the scales. In this case, on the outer part of the ring in the center is the weight in grams, which I use unless the teacher expresses a preference to do the weight in ounces. This first sketch has the needle pointing to 50, and there is a digit 50 on the face of the scale. So I have the students count, starting with the hash mark to the right of the 0, count by 5 so they get to 50, and show them that that's how you read the dial. I then have a second one where the hash mark is at 25, where there is not a digit on the face of the scale. Have them count by 5s from 0. I often have them count as a group till they get to 25 grams. Note that the hash mark at the 25 gram mark is a little bit longer than most of them because it's halfway between 0 and 50. Then I'll have them do one to the 30 gram mark, which is a regular size hash mark as opposed to the longer hash marks at the 25 gram mark in multiples. And excuse me, finally I'll have them do one in between 2 of the hash marks and interpolate, since the hash marks are every 5 grams, interpolate that this one is 33 grams. The class is broken out into groups of 4 or 5 students. Each group has a scale, and each of them has a set of objects. So I'll have each student in each group weigh an object. I bring 5 C-size batteries, 5 golf balls, 5 baseballs, and I'll have the students, this first student in each group, weigh one of these objects, record it on a data sheet. I teach them how to calibrate the scales using the dial in the back. So that's one of our vocabulary words as well. There's the golf ball. Calibrate so it reads 0 at the beginning. Explain that often scientific equipment has to be calibrated before it's used. And the students will record the weights of those 3 objects. 3 different students in each group will weigh one. So you want every student in the group to have a chance to weigh one object. Then after these 3 objects are weighed, I'll have the next student take a plant. These are bamboo plants. You can order them online. I think it's buyluckybamboo.com. And they'll dry it off and they'll weigh it on the scales. And I'll calibrate the scales first. Try to make sure that neither the roots nor the leaves are touching the desk. So I'll have them put the scales on the corner of the desk. And then they will record the weight of that plant on the data sheet. And then I collect those plants, say there are 5 groups from all of them. We add up the grams of those 5 plants, and that'll be the baseline weight for the plant biomass that is going into this container that does not have a betta fish in it. So this container is getting no nutrients for the duration of the experiment. I try to make sure these plants are nutrient deprived at the beginning by keeping them in tap water for, first of all, washing the roots and keeping them in tap water for up to a month ahead of time, actually, if you have the luxury of time. And so we'll add those 5 grams, and I'll have that first set of plants from all the groups placed into this container without any nutrients. Then I'll repeat the procedure, having each group weigh another plant, and again they'll record it on the data sheet. This will be another student will weigh that, record it on the data sheet, and add those 4 or 5 numbers, and that'll be the baseline biomass weight for this container that has the fish in it. And we'll put all 4 or 5 of the plants in that one. The students then feed the fish each day 6 of these betta pellets, 18 on Friday, and I have a data sheet that they record that on. I find it's useful to get the students used to recording data and to make sure that the fish neither get underfed nor overfed. This data sheet has the date, the number of pellets, the initials of the feeder, and then any comments on the condition of the plant, the water, the fish. I change out about 1/3 of this water in the container with the fish each week so it doesn't go anoxic and kill the fish. We also have to make sure this water is not chlorinated, and if you're on well water you don't have to worry about that. If you're on tap water, you'll either have to let it sit for 3 days before you put the fish in it, or you'll have to use dechlor chemical that's available at pet stores as well. Let me just see if there's anything else to cover. I'm going to do a separate video on the second session. These scales are fragile. Always have to make clear that to the students. Here's a digital scale. I can use that to double check the weight, the combined weight of the 5 plants that go in here before it goes in, and the same on that. Just because reading the scales is a challenge for many of these students, and that'll give you a little bit of a check on whether it's accurate or not. As we start the experiment, I asked the students to guess what might happen, which plants might grow more, and of course the hypothesis that I'm hoping will come true here is that these plants will grow more because they get nutrients. Another hypothesis that students make, which is very good, is that they think the fish is going to eat the plant, or nibble on it, the roots in the plant, and therefore that's not going to gain weight during the experiment. So that's another good hypothesis. You can, if you haven't already, you can introduce the term hypothesis. I usually use that for another experiment just because I do try to limit the number of new vocabulary words I introduce each week. At each session I review the vocabulary words from the previous session, and then I introduce the ones that we'll be getting to that day. So that's part 1 of this experiment, and I'll get to another video on part 2, which is generally my last session with that class 6 to 8 weeks later.